All right. Hi, my name is Jacob Sprock. So the whole idea of our research is based around this idea of commensal yeasts. So these are yeasts that live in and on our bodies but don't necessarily hurt or harm us. So we don't really know how these yeasts spread, which is the problem. We want to know like why certain people get these yeast, like why it spreads, or like how it spreads from person to person among like certain groups, maybe like football teams, maybe in certain dorms, if it's on like physical things that people touch, that sort of stuff. So when choosing a yeast that we want to study, we wanted to pick a very common one. So we picked Candida albicans, which among the yeasts that are in your mouth makes up about 80% of those that have been swabbed, right? So one more interesting thing about Candida albicans that we are studying is the fact that it is an opportunistic pathogen. So that means that it's going to lie dormant whenever like things are normal and you're okay. But among people who are immunocompromised, who are like infants, who are elderly, it will attack. It will infect. It'll cause these oral thrushes, which you may have heard in infants. That's Candida albicans. So learning like how this spreads, learning why certain people get it, maybe learning specific like strains of Candida albicans that spread more often can help us prevent people from getting it and from getting sick. So Blake can explain some of the materials and methods.
Hi, I'm Blake. So I'm going to talk about the materials and methods. So what we did was we had students self-administer oral swabs. So they would swab their mouth with a sterile cotton swab. And I think Christian has an example for you. Yep. So they would swab their mouths and then they would spread it on a plate like this one. So this plate is a Sabro medium with chloroamphenicol. And the reason why we chose that plate was because the Sabro medium, that supports the growth of yeast, but the chloroamphenicol, an antibiotic, it stops the growth of bacteria. So that way we were isolating just yeast growth if that person was carrying yeast. So from there what we did was we transferred those to Candida, Chrome agar Candida, which supports the growth of Candida. And the reason why we chose that specific agar is because between different species of Candida, they show different colors on this medium. So according to our interpretation chart here, green colonies, for example, which was the most common, that's Candida albicans. I think Christian has an example of what they might look like in real time.
Yeah, so this is Candida albicans on this side, this green, and then this is another species of Candida on this side, the purple. But so we'd streak each side of the plate. So this is one person and this is another person's yeast, and
that's pretty good. Meagan can tell you a little bit about our results that we found this fall.
Hi, I'm Meagan. So what we found this fall was that the people that did carry Candida, we found that most of it was Candida albicans. So you can see in our chart here that the blue shows that people that didn't carry it, but the red and yellow is who carried it, and you can see that majority was Candida albicans, which is the same as like research that was previously done. So we found that, and then we also carried them from 2 semesters, so we were interested in what could have changed between semesters. So the people that we were able to swab twice, we found that some people that had it the first time, they didn't have it the second time, and that was about a quarter of the people, as you can see in the charts up there. And then we were also looking at people that didn't have it the first time and then they gained it the second time, which was interesting to us because we want to know how they're picking it up, where are they getting it, what trends we can find. But right now our sample size is pretty small, so we don't know, we can't draw any conclusions yet, but we hope in the future that we can, and Blake can tell you about what we plan to do.
Yeah. So yeah, like Meagan said, we are going to add more samples in the future so we can kind of study trends and transmission rates. But what we really want to do, like for right now, all we can really tell is, does someone have Candida albicans or do they have Candida tropicalis or any of the other species of Candida? What we want to do is look at the DNA. We want to extract DNA and isolate it, and then we want to look at the genomic differences and see, okay, within the samples that are like Candida albicans, maybe certain people carry specific strains of Candida albicans. So maybe everybody on the 5th floor of Pad Hall, all of them have the exact same strain, or maybe they don't. We don't really know. So the reason why this is important kind of goes back to what Jacob started with, is that it's an opportunistic pathogen. Recently in the news, there's been another species of Candida, Candida auris. It's becoming prevalent in the clinical setting, causing disease. So with our data, looking at specific strains and how they transmit over time, we might be able to predict when somebody may start carrying a specific strain of Candida. So maybe a certain hospital has a lot of Candida auris, and that's been causing a lot of trouble with babies with oral thrush. Maybe we'll have those babies born at a different hospital until we can sterilize the environment and save lives and stop disease from occurring. So that's our overall goal. So right now we're going to continue collecting samples and isolate DNA and find specific strains of Candida. So.
And if you want to try self-swabbing at home, Christian can also walk you through the process of
I would
swabbing.
say we are going to observe since we put the boxes out.
Yeah, so,
Okay.
so if you want to take it home, so all you do is you swab for about 30 seconds in your mouth, just like cheeks, tongue, everything, and then you open it up and you spread it back and forth on the plate, and then close it back up and wait about a day or two. And if you are positive, it'll show up like this white right here.
That was about a third of the people that we
And then I should